Welcome to class. I will still remain your facilitator, Alawudi Tunde, and because it's CRP305, crop genetics and breeding. We started this course last week, and I started by defining some terms. For those that were not in class, they were not in the last class. You can always check the recording. You check your portal, you see the facilitation recording. Because of time, I will not go back to what I did last week, but we started the course last week. So you are joining today. You are not too late. You're welcome. So the course, like I said, is crop genetics and breeding. And my name is Alawudi. Today my email is yalawudi@noun.edu.ng. So you can feel free to send any message to me if you are not clear on anything that anything you read. So without wasting much time, let us talk about genes. Okay, so today we are going to talk majorly about genes, because you cannot talk of genetics without mentioning genes. So what are these genes? Okay, genes are unit of hereditary. They are hereditary units, and they are transmitted from parents to offspring, from one generation to the next. So these genes are very strong, and they are transmitted, they are generated. So you are a product of your parents. That is what genes is about. As it is in human beings, we also have them in plants. A baby or a child is always a product of the parents. The parents, when they come together, the genes mix. So they mix, and then it forms a child, a baby. And that is why if most cases, when they want to, if they doubt the paternity or maternity of a child, they always go after the DNA. That is where the genes are. The genes are situated, they reside on a long molecule called the DNA. And the full meaning of DNA is deoxyribonucleic acid. So you can see an illustration here. We have the chromosome. This chromosome is found on the DNA. The DNA is usually long like this. It's a double helix bond. The DNA, this course generally is more of abstract, because most of the things that happen on a gene level, and genes are not something you can see with your naked eyes, but you can always see the product of your genes. You can see the product of your genes in your genotype. For those of you that were in the last class, we talked about, we said that the phenotype of a plant or an individual is as a result of the mixture of the genotype and the interaction, genotype, environment, and interaction of genotype by environment. So these genes are not what you can see with your naked eyes ordinarily, but you can see the effect in the phenotype. That's the physical product. So I will try to be as clear as possible, because this is an introductory class. It's just introduction to crop genetics and breeding. Is quite complex, but I'm trying to simplify it for you. So you have the chromosome here. The chromosome is found the genes are found on the chromosome and they reside in the DNA. Okay, so let's move on. This is just a note saying that it's not so clear. I downloaded it. A gene is a section of the DNA, like you saw in the other picture. It's a section of the DNA on a chromosome that codes the information of a protein controlling a specific characteristic of organism. Okay, so the gene for tallness, if you as an individual, if you have parents that are tall, then definitely you are carrying a gene for tallness. Now it depends on the form in which your parents are carrying it. If your parents are carrying it, like we will talk about later, in the homozygous form, your mom is carrying the gene for tallness in the homozygous form, your dad is carrying a gene for tallness in the homozygous form, then you as a child will definitely be tall because you will be carrying it also. You have the genes of your parents. So that is what they are trying to say. Because the genes are in the DNA, they will always control certain characters in you or in the plant, as the case may be. So the gene for tallness will give instructions to the plant cells to generate many plant growth hormones and produce protein due to which the plant will grow tall. So it's like sending an information, the DNA. If it has that gene for tallness in you, to send the information to the brain, and then the brain will start, just keep producing genes for tallness, and you just see that all the plants will grow tall. So we want to talk about gene symbols and terminology. There are some terms that are used in plant genetics. There are some terms that are used in genetics, and we want to talk about some of them. We talked about the DNA, that it is a protein matrix and it forms nucleoproteins, which becomes organized into structures and properties called chromosomes. So like I said before, the DNA resides in the chromosome and it is found in the nucleus of the cell. It's important to know. That's why in the first lecture, I showed you what a plant cell looks like. So the nucleus is that thing in the middle. That is where all the genetic materials, that is where genetic activity takes place, in the nucleus of the cell. Okay, so letters of alphabets were used, like we will see later in class, to denote the symbols. You have to use letters of the alphabet so that you will not be confused. I will show you as time goes on. We'll see and we'll be able to use it. Because I hope you have your pen and your paper, because you try some crosses out by yourself so you understand better. So you use letters of the alphabet. You may use A, B, whatever, depending on what you are representing. Okay, so moving on, this gene has characteristics. Genes have characteristics, and the first one is that genes are hereditary units. They are hereditary and they are transmitted from parents to offspring. That is one characteristic of a gene. It is transmitted from parents to offspring, from one generation to the next. It's important to note they are hereditary. That means that you can inherit them. Genes are inherited. They don't just come there. They are here, actually inherited. Some people even inherit sicknesses because the sickness is inside the gene of their parents, like high blood pressure, sometimes some cancer, some forms of cancer. That's why they want to treat some patients in the hospital. They tell you to bring your family history. They want to know the history of your family. They want to know any member of the family that died of cancer or is going through such things, because it is inherited, it's transferred. is transmitted from parent to offspring. Now, number 2 characteristic is that the genes reside in the DNA, like I talked about. That is where you find them. You find genes in the DNA, and the full meaning of DNA is deoxyribonucleic acid. So, moving on, other characteristics is that DNA plus protein makes up the chromosome. That chromosome that looks like X, in some cases it looks like Y. You have the X chromosome, the Y chromosome. They are made up of proteins, okay? And these chromosomes are also found in the nucleus of the cell. So number 4 is the behavior of these genes is parallel to the chromosome. When you say something is parallel, they don't meet. So the behavior of genes is not the same as the behavior of chromosomes. They are parallel, they are apart to each other, they are parallel. So number 5 is each gene occupies a specific position on the chromosome called the gene locus. That position on the chromosome where the gene resides is called the locus, and the plural of locus is loci. So all these genes on a single chromosome, all the genes on a single chromosome is called a linkage group. And wherever the chromosome goes, of course, it carries all the genes because they are all, it's just like a rope. All the genes are attached to it like that. So wherever the chromosome goes, it carries its genes along, and they form this linkage group. Okay, and linked genes are not transmitted independently. So it's just saying the same thing. Because the genes go along with the chromosome, they cannot work on their own. Okay, so they work together. And number 8, genes in different linkage groups, that's the genes on different chromosomes, are transmitted differently because they are not on the same. If they are on the same chromosome, they are transmitted together. If they are in different chromosomes, they are transmitted differently. So these are the characteristics of genes. It's important that you learn at least 5 of them. At least you should be able to know at least 5 of them, even if you don't know all. So those are the gene characteristics. And now talking about the terminology. Let's move on. The first terminology we'll be talking about here is the phenotype. The phenotype, like we all know, that's the external appearance. Because when I tell you to define some things, I want to see some key points there. Because all of us are scientists, so we should be able to speak like scientists. If I say, what is a phenotype? You should be able to tell me is the external appearance of an individual or plant that makes it different or distinguishes it from other species of plants. When you see maize, why is it that you say this is a maize plant and you don't call it cowpea? Because of the physical appearance, because of what you can see, that is the phenotype. What you actually see with your eyes. You see a plant on the field that you say, oh, this is maize. Oh, this is cowpea. Oh, this is vegetable. Oh, this is yam. Because of the physical thing that you see. So that physical thing you are seeing is the phenotype. So, and this phenotype is determined by, like I talked about in the first class, is determined by the genetic constitution, its environment, and the interaction. I gave you a formula there. The phenotype, VP equals to VG plus VE plus VGE. Make sure you check it. It's inside your course material too. So that is the phenotype. Now, talking about the genotype. You know, the phenotype is what you can see, but the genotype, you cannot see it, but it is there. Okay, So genotype is the genes, all genes possessed by an individual. That is what the genotype is. The genotype is all the genes that are possessed by an individual. And that genotype is what is inherited. That's what you inherit from your parents, from your generation. It's passed down. It's passed down. It may be like in this picture now, it may be the color of the eyes. In some families, the color of their eyes is blue. In some, the color of their eyes is brown. In some, the color of their hair, it could be the color of their hair. So these are genes. They are passed down from gen. It could be the color of the skin. If your parents are white, you cannot be black. It's just your genotype, because your parents, all their family, they are white. So definitely you coming out black, there's an issue. If a plant is tall, if the parents are tall, the plant will definitely be tall, because the plant has that gene for tallness. So the genes possessed by an individual is the genotype. Now we are now going deeper, and we are talking about homozygous. You know what phenotype is, you know what genotype is. Now, homozygous is talking about the union of gametes. Gametes is, like I talked about in the first class, gametes are cells, okay? So if the gametes are identical, they are carrying identical allele. Allele is just what represents the gene, okay? The representation of the gene, that's what the allele means. Now you can see in this picture here that the allele for this gene is capital A, capital A. If it is capital like this, or it is small a like this, that is what we are using to represent the gene. You know, we said that we'll be using letters to represent so that you will not be confused. So wherever you see the allele in capital form like this, it is homozygous. That means it is identical. Homozygosity talks about being identical. You can see here that is capital A, capital L. So this is an allele that is homozygous. Also, small letter, you won't say because it's small letter is not homozygous. It's small letter, small a. So this one too is carrying an homozygous allele. But if you look at this third one here, you can see that it's capital A and small a. So you can't call that homozygous. That is what they call heterozygous. That means they are carrying different alleles. So if you see an organism or the union of gametes that involves identical alleles, then that is producing a homozygous genotype. But if the alleles are not identical, then it is a heterozygous genotype. So I'm still trying to illustrate here. The female, of course, that is from the female that you get the egg, and it's from the male that you get the sperm. So what the female is donating here is capital A. What the male too is donating from his body is capital A. Now, when fertilization takes place, these alleles, they come together, they mix together, and the gamete that is formed, or the child that is produced, or the plant that is produced, we carry identical alleles from the parents to form capital A for capital A. So that is what is produced. Now, if you want to say what gamete is this produced? You don't need to be carrying the two A's again. You just have to represent it with one, except it is different. If it is different, you have to carry the two. But carrying the two here, when you want to talk about the gamete, you just produce one. So this is an illustration of homozygosity. And it's important to note that in crosses, the female parent is always written first, like you can see in the picture before. The female always comes first. If you want to do your crosses, note that the female comes first. That's the female is written on the left-hand side, and the male is on the right. That is how we do our crosses. And the homozygote genotype produces one kind of gamete. Like I said here, it is capital A, capital A. So if I tell you what gamete does this individual produce, you just have to give me just one, A. But if it is different, then you have to write the gamete there. So that is homozygosity. Now, in pure lines, okay, pure lines are usually homozygous in nature because they are pure. They are not contaminated. You can't see any other kind of mixed gene. They are pure. So a group of individuals with similar genetic background is often referred to as a pure line. This is a mistake. Pure line or variety. So when you have similar genetic background, it's a pure line. And the matings, you can always get pure line by mating homozygous individuals. I showed you what homozygous means. I have shown you what the crossing is like. So mating between homozygous individuals will always produce a pure line, and the offspring is exactly like the parents. Exactly, no difference. If you see them on the field, you can't even differentiate them because they are exactly the same thing. So pure lines are plants that show the same traits generation after generation. Nothing changes in them. If you plant a pure line, if you plant it 1,000 times, you get the same result because it is pure. It doesn't have anything else that is contaminated in it. If you plant it 1,000 times, you get the same thing. So from generation to generation, that is how it just keep multiplying and just keep producing itself. And Mendel, we'll talk about Mendel later. Mendel is the father of genetics. He's the one that introduced this topic to the whole world. So he's the father of genetics, and that is what he used for his first experiment. We will talk about the experiment later. So that is what a pure line is about. And pure line means those breeds of organisms that have been generated through repeated self-fertilization. When you begin to self-implant continuously for 7 or 8 generations, you get a pure line because there's nothing else to cross again. So you get a pure line, and the trait is passed to the progeny if bred with another true breeding organism. Pure line. So we are moving to another one, heterozygous. Like I explained the other time, heterozygosity or heterozygous is the union of gametes with different alleles. Different alleles. In the case of homozygous, the alleles are identical, they are the same. But heterozygous now, they are different. That's the meaning of hetero, different. Homo means the same. So in this case, different kind of gametes are produced. In the case of homozygous, you saw that the gamete produces the same, but heterozygosity produces different kind of gametes during gametogenesis. I explained that process in the first class. So like you can see here, The genitive describes the genitive makeup, blah, blah, blah. Now, see this capital B, you all know that it is called homozygous. Homozygous because it is the same letter. Dominant because it is capital. What a dominant gene means is that, let me put it this way. Let me give an illustration. Because genes are competitive in nature. They always want to express themselves in the phenotype. They always fight so that people will see them. So there are so many genes inside you, but it's not everything that will show in the phenotype of an individual. So there are the genes that show, that you see. For example, let me use the color of flowers. You know that every plant has its flower. There are some plants that are different, that have different colors of flowers. For example, if a flower is white, you see that the flower is white. That white color is dominant because you can see it, that this flower is white. In some cases, you will see the flower yellow. That color is dominant. We'll still talk about it later, but I just want you to know when to be able to use capital letter and small letter. So if the gene is dominant, you represent it with capital letter. If there are some genes that are homozygous dominant, you represent it with capital B like this. If it is heterozygous dominant, because it is hetero, it cannot be both capital. It has to be capital B and small b. And if it is homozygous recessive, this homozygous recessive, they are not dominant, they are recessive, but you see them, they come just like a flash. They are there, but you won't see them on time. When they represent themselves, it is kind of late. As we go on, you will understand. So you know what heterozygous is and homozygous now. And over here, types of an organism having two different alleles for a trait, that is heterozygous. And the two different alleles, one is usually dominant and one is recessive, like you can see here. A capital B, like I said, is capital because it is dominant. It is small letter because it is recessive. So a mixture of this is heterozygous. So one is usually dominant and one is recessive. Represented in genetics by having one uppercase and lowercase. For example, capital B, small b, and brown eyes, and recessive for blue eyes. Yes, okay, let me explain this. If you say that a mother, the mother of a child has blue eyes and the father has brown eyes and they have a baby. The mother has blue eyes, the father has brown eyes. They now have a baby. And the baby has brown eyes. Okay. That brown eyes in that baby is in the heterozygous form. Okay. It's in the heterozygous form. That means that brown eyes is dominant over the blue eyes of the mother. I hope you are getting me. The brown eyes is dominant over the blue eyes. That is why you can see that the baby has brown eyes. So the fact that that baby has brown eyes does not mean that the baby does not have the gene for the blue eyes inside, but that gene is sleeping, is inside that baby. but that gene is not showing in the physical form. It's inside. So it is recessive. It is sleeping. It is not outside. It's just laying there inside. The thing that will make that baby to have blue eyes is if that baby grows up and then marries somebody again with blue eyes. So that blue eyes that is sleeping in that baby, in that child, as it were, when it fertilizes again, mixes again with the blue eyes of his wife, they will now give birth to one child out of maybe 4 or 5. One of their children will have blue eyes. So that is just the best way I can explain this recessive and dominant gene for. That dominance is just like something dominating over something. Is the one that is showing itself. That doesn't mean that the gene is not there or is recessive. The dominant one is the one that we will see. So let's move on. This is homozygous versus heterozygous, like I've explained. You can see capital B here, capital B. That is the dominant gene, heterozygous dominant. This is small letter B, small letter. This is homozygous dominant, sorry. And this small letter B, small letter D is homozygous recessive. Now, if you cross both of them, this is what you get. You get heterozygous individuals. Okay, I've already discussed this. So the difference between homozygous and heterozygous in the genotype. 2 identical alleles in a gene for homozygous, 2 different alleles for the same gene in heterozygous. Then in the phenotype, that's what you can see. An observable trait from 2 identical genes. And in the case of this, for the heterozygous, the organism possesses 2 distinct alleles. However, it's the dominant one that will be expressed. Now, let's talk about Mendel's experiment. You will understand clearer when we discuss Mendel's experiment. So, Mendel, like I said, is the father of genetics, and he was just working on his farm. Mendel was working on his farm. He was working with peas. You know what garden peas, these green peas. That was what he was working on. So he just decided, okay, let me just plant these peas. So what he did was that he took peas and then he planted it. He took okay, let me see. From his work, he proposed 2 genetic principles. The first principle is known as the principle of segregation, and the second one is known as the principle of independent assortment. So the use of chi-square calculation will be demonstrated for all this. Okay, so what he did was that he now planted peas. This is his experiment. He crossed a tall plant with a dwarf plant. He was using sweet peas. So the tall plant, let us say, is demonstrated by capital T, and the short one is will be represented by small t. So there's a whiteboard here. There's a whiteboard. Let me see. Sorry for my board here. Okay. So what he did was to, I hope you can see my board.
Yes, we
Is
can see it.
anybody there?
Yes, ma'am, we can see.
Okay.
Yes,
So,
ma'am.
okay. So we are representing the tall plants with capital T, and then the short one with small letter t. So this is what we have. We crossed it. That's the cross. So who can now tell me the garment that was, what is the garment that is produced for this capital T, capital T? What garment is produced there? Who can tell me?
Homozygous.
You represent, because this is two capital, so the garment produced there is just one small t, okay, and one t, and the garment produced in this one for the small t is just one of this. Hope it is clear. So when he crossed it now, this was his result. This was his result. Hope you understand. This is homozygous for tall. This is homozygous for short. So the gamete produced is this, and the gamete produced here is this. So when fertilization took place, this was what Mendel saw. So all the plants were tall. Okay? Hope you understand. All the plants were tall. So what he now did was that he now took this zygote, this baby, this thing that was produced, and he now selved it. That means he mated it with self. In plants, you can do that. You can mate plants to themselves. But in the case of human beings, you cannot, they don't encourage mating to oneself in human beings. And I will tell you the reason why. Incest is not good because as you are like this, you cannot marry your brother and your sister, because the results that you get, please mute your mics. Somebody's mic is disturbing. Okay, because the results you will get is very, that you will see all manner of rubbish. That's why it's not encouraged to do it in human beings. We have a lot of genes inside us. Some are good genes, some are bad genes. So if you mate a human being to himself, if you marry your sister or your brother or even your father, the gene that is not good is the one that will come out. But in the case of plants, you can mate plants to themselves. As a matter of fact, they encourage it in plants and animals. If you want to maintain a pure life, you mate to themselves. So what Mendel did was that he mated this plant, this capital T, small t, he mated it with self. So this is what he got. This is capital T, small t, times another capital T and another small t. So the gamete that is produced in this case, you have the capital T gamete and you have small t gamete. Those are the gametes that are produced in this case. And then another capital T gamete and small t gametes. So when you now cross them together, When you cross it together, okay, when you cross this and this together, hope you can see it. The result you get here is when you cross this, let me take this straight line. When you cross this to this, because you have to cross them to each other, what you get hope you can see it. Then you cross this, so this was what the man got. Somebody sent me a chat. Okay. So what he discovered is that he saw three tall, because this one is twice. Let me do it for you. What he discovered is that he saw three tall plants and one short plant. He was now wondering, where did the short plant come from? That was the beginning of genetics. He was now wondering, how can I plant a tall plant and then get a short plant? This is short because it is homozygous recessive. It's smarty. So this plant was short. Why these three other plants are still tall? That was what Mendel discovered. Hope that is clear. Who doesn't understand? Okay. Let me go back to the PowerPoint slide. So he crossed a tall plant with a dwarf one of sweet pea by placing the pollen. The pollen is the male. So you put the pollen, which is the male, on the stigma of a dwarf one, which is the female. So the female was the dwarf one, the male was the tall one. And the F1 means the first filial generation. That's the children. Let me put it in that way, in a layman's English. The children of the parents. Those are the F1. Okay. So the F1 that he got, all of them were tall. Then you now selfed it. When you now selfed it, that's the F1, it mated F1 to itself. That's the meaning of selfing. When you talk about selfing, you are meeting to itself. So he now got his F2 because he made the F1, the tall one, and the tall one to be the parents. That's capital T, small t. So at the end of the day, he now got a ratio of 3:1, 3 tall to 1 dwarf. He was now wondering that where did this dwarf come from? So he now went later, he now realized that the gene for dwarfness is inside those tall plants. So it's when he selfed it that he now saw that that short one was there. So he now decided to do a lot of illustrations using seed shape. In some cases is round, some cases is wrinkled. For the same sweet pea, seed color, in some cases the color of the seed is yellow, some they are green. He just started doing a lot of mating, pod shape, some inflated, some constricted, flower color, some are purple, some are white, pod color, some green, some yellow, plant size, some are tall, some are dark. So it now, all these traits, because they call them traits, they are called characteristics in genetics. Mendel now started to work on all those traits. He now started getting all manner of results. That is how genetics came into being. And genetics is used by a lot of plant breeders, because today, if you are looking for a better plant, you cannot get a better plant if you do not manipulate the genes of that plant. Genetics is all about manipulation. You manipulate all those genes in every form, that at the end of the day, you will get the result you are looking for. If you are working on cowpea, for instance, and you want a cowpea that is producing a high yield, and you cannot find it, as a breeder, you can make it. That is just the power that breeders have. They can make things by themselves by manipulating genes through genetics. And that is the only way to go. So what they do is that they will take another cowpea. There are some cowpea that they are producing high, but their problem is that they easily fall, they easily have diseases and pests ravaging them. So what the breeder will do is that he will go to the lab, go to the genes of that plant and extract the good ones by crossing. We will talk about it later. There are a lot of methods that you can use: backcross method, progeny testing, mass selection. They will do all that and manipulate the genes. So at the end of the day, they will get a plant that is high yielding and resistant to diseases. So that is why genetics is important. So in summary now, from what we did, the phenotype, like we talked about, is what you can see: tall and short. At least if you see a tall plant, you will know that this plant is tall. If you see a short one, you will know that it is short. So that is the phenotype, what you can see. In the case of the genotype, what you cannot see, that's what we use letters to denote. The tall one, we denoted it with capital T, that is homozygous tall. In the case of heterozygous tall, it is tall also, but it's in the heterozygous form. That is capital T, small t. That is heterozygous tall. And short dwarf, this is a mistake. The short one is supposed to be small t, small t. Sorry, this is a mistake. I will correct it. It's small t, short. Small t, small t. Now, in the ratio that you saw from what we did, the ratio, the genotypic frequency, or the ratio, we had one tall plant from what I crossed on the whiteboard. Let me see if it is still there. It's not there again. Okay, it's still there. From what is on the whiteboard, the ratio, you can see this is one tall, this is another tall, this is another tall. So you have ratio of 3 to 1, one short. So that is ratio 3 to 1. That's the phenotypic ratio. Okay, so the genotypic frequency is 3 to 1 to, yes, the genotypic frequency, please listen very well, is ratio 1 to 2 to 1. 1 for the big T, 2 for the heterozygous, that's big T, small t, big T, small t, we saw 2 of it, and 1 of the short one. And the phenotypic ratio is ratio 3 to 1. So please note that the genotypic ratio was ratio 1 to 2 to 1. Hope you knew how that 1 to 2 to 1 came about. While the phenotypic ratio was ratio 3 to 1, what you can see, what the man saw was 3 tall plants and 1 short one. But the genotype is saying that it's only 1 tall one, homozygous tall, 2 heterozygous tall, and 1 homozygous short. So you can always practice. You can always practice. These are Mendel's principle. These ones you should know them. You can know them often if you read it. Principle of segregation states that in any parent, only one allelic form of gene is transmitted through a gamete to the offspring. And in the case of principle for independent assortment, it states that the segregation of one factor pairs occurs independently of any other. These are things that you can read up yourself. Okay, because time is going. I don't want to waste your time. So let's quickly talk about monohybrid crosses. Mono stands for 1. Hybrid is the product of a cross. So when you are talking of monohybrid cross, is the cross in which only one single pair of allele is considered. One single pair of allele is considered a monohybrid cross. For example, if you want to, like what we were doing before, if you want to look at tallness alone, tallness of a plant alone, that is just one factor. Tallness alone is monohybrid. So in this case now, this sign over here is the sign for male. This sign over here is the sign for male. This sign over here, like cross, like this, is the sign for female. Okay, that's the genetic sign for female. This one is the sign for male. So in a monohybrid cross now, they are telling you that the male is carrying this allele and the female is carrying this allele. These are the offspring that will be formed in a monohybrid cross. So let me just quickly show you on the whiteboard again, because I'm going to give you an assignment too, and you will bring it to the next class. You come and, in fact, I'm not even going to, one of you, let me even call somebody to do this for me. Madam, I don't know if you are using your son's number, because I heard a woman's voice in this Adeni job. You were in class when I was talking about it. So please, if you are using your laptop, you can join me on this page, on this whiteboard. I want you to come and illustrate that monohybrid cross. Adeni job.
Okay.
Are you there?
Yes, ma.
Okay.
I'm a man. Oh.
Okay.
My voice just looks like that of woman.
Oh, really? Sorry. You're welcome. Okay, sorry about that. Okay, you can join me on the board. We want to
Okay.
we want to demonstrate monohybrid cross, right? And
Okay.
there's an illustration there. You have the male as capital B, small b, and the female as capital B, small b. So come and show us how to do it, how to cross it.
Oh, but I'm using a phone, ma.
You're using a phone.
Yes.
Okay. Who is using a laptop? Musa Farouk, you used to be on a laptop. Are you there?
Yes, I am here, ma'am.
Uh-huh.
I have a laptop, but I actually log in through my phone. But let me quickly see if I can log in through the laptop.
Okay, please, who is on a laptop? I want somebody on the laptop to join me. I don't want to be the only one doing it. I want this class to be interactive. That's the only way you can get it. That's the only way you can get it.
I am trying to log into the using my laptop.
Gloria, are you on the laptop?
No, ma'am, using a phone.
Wait again, Adinka, laptop. I see you, Joshua. Are you on the laptop?
I'm in the car, so I'm
No,
using
I'm using a phone, ma. Using a phone. I'm not at home. I'm using a phone.
Wow. Wow. What about Oyedoku? Laptop. Who is on the laptop there? I need somebody.
What is this time?
Yeah, Farouk, we are waiting. Let's just do that. So, you're going to.
Just a minute. The network is kind of.
Okay. Don't let me waste time. Don't worry. You are going to snap. You will do the assignment I'll give you, you will do it on a paper, then you will snap the paper and then you will send it to my WhatsApp. Yes.
Hello.
Not now, in the next class. I will give you for the dihybrid cross. I will show you the monohybrid cross, then I will give you for the dihybrid cross. So in this monohybrid cross now, from what is on the slide, you have this cross. You have heterozygous for I think color. Yeah, so for the flower color, we want to demonstrate using monohybrid crosses. So this is what you have, the self-date. So the garment that is produced here is going to be what? Who can give, what's the garment that is here? Who can tell me? You have a capital B garment, and then you have a small b garment. Okay? Those are the garments. Because this one is not the same. The same thing over here, you have capital B garment, and then you have small b garment. So this is what you are going to cross now to each other. Okay? And if you do the crossing and you do it well, you will see that this one will pair with this. That is one baby is formed here. And the baby is capital B, capital B. You can see it here, where I joined it. Another baby is formed here. Okay, let's exhaust this first. another baby is formed like this. Capital B and another one is formed like this. It's not difficult. You just have to pay attention to it. You can see. So another one is formed like this. This is formed like this. That is capital B and small b. And then another one. So you get 4 different types of progeny in this particular selfie. So these are the 4 that you get. Now, who can interpret this result for me? We are talking of yes, I'll give you my WhatsApp number. I even want you to even form a group for yourself. Then you add me. You should be able to have a class rep. Gloria, that maybe you'll be our class rep. You form the group, then you add everybody. Please just drop your number so that she can add you to the group. Because I'll be getting.
Let me.
We'll drop your numbers in the chat session, yes, so that you can form a group. Yes, I can hear you.
The interpretation, ma'am.
Yes, the interpretation, I can listen. I'm listening to you. Yes, what's the interpretation?
The genotypic ratio is.
Let us start with the genotypic. Okay, yes, the genotypic ratio is, yes.
The genotypic ratio is 1 to 2
to 2
to 1
to 1.
That is, we
okay,
have.
so somebody should give me the phenotypic ratio. Somebody else should give me the. Yes, the phenotypic ratio is 3 to 1. So I think you can understand it well like this. The genotypic ratio is 1 to 2 to 1. That means that 1 purple color in the homozygous form, 2 purple also, but is in the heterozygous form. Then 1 white color in the homozygous form. That's the genotype. And for the phenotype, what you can see is 3 purple flowers and 1 white flower. So, Gloria, I hope you can see the numbers there. Please. All
yes, I can see.
You be our class rep for this class. So form a WhatsApp group. Add everybody. When you're through, you can invite me. Let me drop my own number too. Oops.
Okay, ma'am.
So that's my number. Just add me to it. Okay. So let's go back to our class. Let me see. Let me share. So that is what happens in this case. Now, For the dihybrid course, you know I said mono is 1. You are just considering only one trait. In this trait here, it was the color of the flower. Just 1. But in the case of dihybrid, di means 2. So it involves 2 genes for 4 alleles. 2 genes, 4 alleles. So you are taking 2 different traits into consideration, 2 different characteristics into consideration. In this case, round seed and yellow color. You know, when we are talking about the characteristics in Mendel's case, let me see. Let me show you this place. There are shape of the seed shape was round and wrinkled, and the seed color was yellow and green. So now in this dihybrid illustration, they now consider the roundness of the seed and the yellow color as against the wrinkled seed and the green color. So your assignment now is that you should go and make the dihybrid cross for selfing of the round and yellow seed to the green and wrinkled. Hope you understand it. What we used to denote, because I want it to be uniform. Let me get a pen also. Okay. What are you going to use to denote the round seed? That is R. Okay. The round is R, and the wrinkled round is capital R. Wrinkled will now be small r. Okay. Round is capital R. Wrinkled is small r. For the yellow is capital Y because it's the dominant. The capital one is the dominant. So yellow is capital Y, while the green is small y. So I want you to cross yellow. I want you to cross round and yellow seeds to wrinkled and green seeds. You show me in the next class. I want to see everybody doing it. So in the case of tri-hybrid cross, let's quickly talk about it before we round up the class. These ones, you are involving 3 pairs of genes. Tri means 3. So 3 pairs of genes with contrasting traits. You can see that it's going more complex. As the genes are increasing, then it will be more complex. But you have to know it so that when you see it, you'll be able to differentiate. So for example, now in this case, they are considering 3 traits. 3 traits means 6 alleles. Okay, 3 traits means 6 alleles, like you can see here. So the first trait is the capital A, capital B. The second trait is the capital B, capital B, and the third one is the capital C, capital C. Okay, so they now crossed it between small a, small b, small b, and small c individuals. Okay, their F1 is ABC in capital and small form mixed together. But the F2 now, this is their F1 here. Okay, this is the result for the F1. But the F2, when you now self this is what you get. It looks complex, so but it's very simple if you get the principle. So first of all, do that assignment first. We'll meet in the next class. You can try and practice the tri-hybrid one too. Try it. There's nothing wrong in it. There's nothing wrong in trying. Try it. Let me see. If you have any issue with it, let me know. Just try it. So this is what you get. Try it. Don't look at what is there. Just try it on your own. These are the gametes. Cross them. That's why they put them in a punnett triangle. Like this is a triangle, so that it will be easier. You can see over here. Let me use my pointer. You can see here that they are crossing the one up here to the one by the side here. If you look at this first box here, this is what you have. ABC capital times ABC capital. That is what you have here. This second box, this is what you have there. When you multiply this A capital, B capital, C small letter to this is what you get. So that is how they got all these gametes. You can try it and let me see. So having said that, I think we have come to the end of this class. Any questions? In the next class, we'll be talking about
how do we, sorry, ma, how do we submit? Are we going to jot this down on our?
You snap it and drop it on the WhatsApp.
Send it to the. All right. Thank you very much.
Yes.
Thank you very much.
Snap it, drop it on the WhatsApp page.
All right. Thank you.
Or send it to me personally on my WhatsApp.
All right. Thank you. Glory, please try and create that group.
Yes.
I hope she has all our numbers.
I know.
Yes, I will do that after the class.
I know you are responsible. You won't put everybody. You won't disappoint us. So. Okay.
Just check the participants.
Any question? Do you understand it? Is there any question? Just go and practice.
Miss Matt, there's one question I want to ask.
Okay.
My question is based on the recessive, based on the explanation you gave earlier that you said if someone has a blue eyes and a brown eyes, if they give birth, one of the child has a brown eyes, but the blue eyes is dormant, a bit recessive, that is sleeping. But if the person now marries someone that has blue eyes, that is when they will not give birth to one of the child that will have blue eyes. But what if the person does not marry someone that has blue eyes? Won't the blue eyes come back?
Sleeping. It will still be there, but it will not until you see another gene to join that to be able to come out. That's what happens in the case of albinism. You see that the parents are black and they give birth to albino. They will now start wondering, where did this albino come from? But the parents are carrying the genes of albinism. They don't know. The gene is inside them. The gene is sleeping inside them there. If they go and check their family lineage, you will see that there's albino in their family. But you know, when we want to marry these days, we just marry without doing history, without doing proper, what do they call, historic background. If you want to know some things, you go to the history and you see things. You see different families with different, all manner of things. That's why incest is not supported. You don't marry your clan. You don't marry your blood lineage, because if you marry your blood, they are going to all the dirty things inside your genes is the one you will be seeing. But it's better for the gene to be there and be sleeping than for it to come out when you start marrying yourselves. That's what happens in the case of selfie. So the parents, they have the gene for albino, but that gene is sleeping inside them. So both of them now meet each other and they marry each other. And both of them have the gene there sleeping. They now give birth to children with albino. The same thing with sickle cell. That is why when you want to marry, you have to do a blood test to know your genotype. Sickle cell, and in fact, those children, I used to pity them. It's not supposed to be. If you do correct genotype testing, you see that you have you have a sickle cell in your blood. You don't have to marry somebody that has sickle cell again for any reason. because you will definitely give birth to a sickle cell child. The gene is there, is sleeping. When he sees another gene again and join, you will just have children that have problems. And the funniest thing about this ratio is that they are saying ratio 1:3. That one, as small as it is, it may be the first 4 children that one has that will just be having sickle cell. That ratio, you cannot say where the ratio will be. The ratio is there, but you cannot tell where it will be. So genetics is very interesting. Very interesting. Okay, somebody said the assignment question again. The assignment question again is that you should cross okay, let me give you you cross a round and yellow seed, round and yellow, okay, to green and wrinkled. You get your F1, you cross a round and yellow to green and wrinkled. You get your F1, then you self it. That's the question. You self it. You get your F1 and you self it. Okay? I will drop the question on your class activity. Check your portal. I'll drop it there again.
Sorry, ma.
Yes.
The donation for these letters, you say capital R is round.
Yes.
Capital Y is yellow.
Yes.
Small r is wrinkle.
Yes.
Small y is green.
Yes.
All right, thank you.
Okay, another question again. Okay, so in the absence of any question, I think it's safe for me to say that the class is over. See you next week.
Thank you.
All right.
All right.
Thank you, ma.
so much.
Please, what's our next topic?
You said?
What is our next topic?
Next topic is chi-square. You do mathematics next
Yes,
week.
ma'am.
We'll also use mathematics next week, so you prepare for maths. It's simple. It's simple.
Okay,
it's simple. You know, I will tell you, it's not difficult. It's simple. But it's mathematics. In fact, I'm supposed to even put it, but I don't want to make the whole class. I don't want it to be too stressful for you. That's why I said let me put it next week.
Okay.
Ma, you said we have another class of the CRP303.
303, yes.
309.
Sorry.
Ma,
yes,
there is
sorry.
no link on the
I sent
Facebook.
another link. I sent another link I sent another link in the morning on Thursday. You didn't get that link on time. Some people still join the class. We have CRP303.
We don't know which course.
CRP303 fraud protection.
Please, I'm not seeing link for that one at all. It's only this one that be seeing link from my email.
Hey, didn't you drop your name?
Yes, ma'am.
Wow.
It's only this one I always receive from my email.
And you are doing CRP303 too?
Yes, ma'am. I even sent you.
Yes,
I even sent you mail.
Yes, Umar,
week.
I saw your message. I saw your message, Farouk. I saw your message. I saw it. Farouk, there was a mistake. It won't happen like that next week. I'm going to send another link again. For you that said that you did not see the link at all, you have to send me your email address, please. You know my email address,
Okay,
right?
Yes,
send your email to me on my email. Please do that now because I'm going to send the link for. Okay, you are not seeing the link too. Okay, Doku.
Okay, ma. Once I create a group, we can.
My email address is yalaude@edu.ng. Please.
Oh, this is our now email address.
Now, this is my email address. Let me put it on my. Send it now. I'm going to send it for CRP now. I'm even supposed to ask you questions on all these terminologies, but I want to assume that you understand them. Because time has gone now. This is my email. Can you see my email address? Please send it to me now, because I'm going to send the link. Oh, no, you can't send it to me like this. So send it to my mail.
Okay, ma.
Send it to my mail. I can't see it here.
Your WhatsApp number too.
Okay, this is my email address.
Please, your WhatsApp number too.
My WhatsApp number, I've already sent it. Is in the chat session. The only thing, the reason why I don't like sending my number is that I don't want people to be calling me and disturbing me. I prefer to do email. But because I want you to submit assignments now, I've dropped my phone number in the link. 003-800-9060. Okay. Have a nice day. Thank you very much.
Thank you.
Thank you, ma'am.
Thank you, ma'am. Good luck.